Hello everyone, my name is Ibrar Ahmed. I am Ph.D. scholar in animal nutrition and nutritional disease in Selcuk University. Today's my topic of presentation is antimicrobial peptides from Hermetia Illucens, which is basically a black soldier fly as an alternative to antibiotics and in animal. Introduction. As we all know, the human population is increasing swiftly, which needs high quality of animal protein. And to get high quality of animal proteins, there is a practice of irrational use of antibiotics, which will lead to drop in meat quality, and it will also create a problem of medicine resistance, antibiotic resistance, and it will also create a problem like humans are also consuming antibiotics and we are also using them in livestock, which will create a competition between humans and animals for the antibiotics. That's why many countries have banned the use of antibiotics. Like in European countries, they have banned the use of antibiotics in livestock. Therefore, the current demand is to find safe alternative antibiotics. One of those alternative to antibiotics is antimicrobial peptide, which is also found in insects. And insects consist of 90% of world planet biomass, and until now only 10% of these antimicrobial peptides from insects has been discovered. And the first AMP from insects was discovered in 1974 from Cecropia moth pupae. Now we will talk about insects, black soldier fly, which is the main topic for presentation. It is considered as an alternative protein source in animal protein, and it has high feed conversion ratio, and it is found in warm weather. And this insect is harmless. It is not involved in transmission of any kind of diseases, and it converts low quality waste product into high quality animal protein source. Now we will talk about different types of AMPs from black soldier fly. There are five different types: cecropins, atacins, deficins, knot-like proteins, and diptericins. On the basis of their structure, there is another classification of insects AMPs. The first one is peptides with alpha helical structure. The second one is high in glycine. Third one is high in proline and arginine, and the fourth one is rich in cysteine. AMPs of BSF. As we all know, because black soldier fly can survive in harsh environment, which typically indicates that it has strong innate immune system, which makes that fly capable to survive in harsh environment. Defensin like peptide 4 is a newly discovered AMP from black soldier fly, and it has been tested in mice. Mice were infested with Staphylococcus aureus, and the defensin like peptide 4 was injected in those infested mice at the rate of 3 to 7.5 mg per kg, and this leads to 80 to 100% more survival rate of infested mice. And this AMP has been also been used in poultry diet as a replacement of antibiotics. In this slide, we will discuss about the mechanism of action of AMPs from black soldier fly. Basically, the main principle is the attack on the cell membrane of the bacteria. The cationic structures of AMPs of black soldier fly, they bind with the anionic lipopolysaccharides, teichoic acid and lipoteichoic acids, and they form ion channels. They break the cell membrane integrity and leading to the leakage of the internal content, and in the final result they kill the bacteria. Basically, they target the lipids present in the cell membrane, and they results in the formation of pore, and this will leads to the disruption of the cell membrane of the bacteria and the death of the bacteria. There are different types of membrane lysis mechanism, but the AMPs of black soldier fly, they follow the carpet-like mode. You can clearly see here the fourth one, the carpet-like model by which AMPs of black soldier fly leads to the lysis of cell membrane of the bacteria. The serotoxin and amphotericin B, these are the examples of AMPs from insects, and they damage the cell membrane of the bacteria. There is another pathway, the AMPs cross the cell membrane spontaneously, and they disrupt the intracellular metabolic processes, leading to death of the bacteria. And some N-terminal region of AMPs, they attach with the DNA domains, specific DNA domains of the bacteria, and they disrupt the transcription, leading to death of the bacteria. The antimicrobial peptide of black soldier fly have beneficial properties like they have better thermal stability, lower molecular weight, broad antibacterial spectra, and unique mode of action. Until now, 57 genes coding for AMPs has been recognized from black soldier fly larvae and adults. Out of 57, 26 encodes for defensins AMPs. In this slide, we will talk about the hemolymph, which is basically obtained from insects, and it acts like antimicrobial peptide. It is removed by two processes: acidic methanol extraction and lipid isolation using chloroform and ethyl acetate. Hemolymph isolates shows great activity against bacteria, like you can see gram-negative bacteria, Helicobacter, Escherichia coli, Salmonella, and gram-positive bacteria like Staphylococcus and Bacillus subtilis, and the yeast Candida albicans. In this slide, we will talk about studies of black soldier fly AMPs. The AMPs of black soldier fly protein meal and chitin powder were evaluated against Clostridium perfringens and Clostridium difficile, and they show positive results. Similarly, the broiler chicks exposed with Salmonella gallinaриum, they were fed different 3 levels of black soldier fly meal and a control diet, and they show a higher immunity in the form of CD4 cells, and the life expectancy of chicks was enhanced in broiler fed black soldier fly diets. Similarly, the beagle dogs which were exposed to E. coli, they were fed defatted black soldier fly meal for 6 weeks at the rate of 1 and 2%, and they showed higher antioxidative and anti-inflammatory capability as compared to dogs fed controlled diets. Black soldier fly prepare meal were used in wean piglet diets as the replacement of soya, fish, and plasma protein, and it leads to no negative impact on their growth performance and serum indices. Similarly, the black soldier fly larvae nourished on kitchen garbage, they were utilized in piglet diets as the replacement of fish and soya meal, and it leads to enhanced immunological capability, growth performance, and nutritional digestibility in weaned piglets. Now we will talk about the conclusion. The black soldier fly derived AMPs have antibacterial properties, and these are new derived AMPs. They have no problem of resistance, bacterial resistance, and they can be a alternative solution to be used as an antibiotic in livestock sector, which is a worldwide problem in livestock sector. And this topic is very new, very novel. There is no more research on this topic. Now we talk about the future direction. There is a lot of gap in this field of study, and these AMP should be studied as an antibiotic replacement, and there should be more identification of pathogens receptors, and there should be clear and deep study of their mechanism of action and investigation of AMPs from black soldier fly as an alternative of antibiotic substances. If you have any questions, you can ask me. Thanks for listening me. Thank you.